Hi, my name is Parisa, and today I'll be talking about lysogenic conversion. So not all viruses reproduce in the same way. Some go through lytic replication and others go through lysogenic replication. Lysogenic replication is a process where temperate phages or lysogenic phages first attach to a bacterial cell such as E. coli, and they eventually manipulate the bacterial cell into slowly replicating genes from their own genome. As I mentioned, the first step is attachment. The virus will basically randomly come into contact with the bacterial cell, such as E. coli, and attach via its tail. The virus then proceeds to inject its own DNA into the bacterial cell, but unlike lytic replication, the host's DNA is not destroyed. In fact, the phage's genome doesn't take immediate control of the cell, and rather it remains inactive for some time. This dormant-like phage is also known as a prophage. This prophage remains inactive by suppressing its own genes, and by doing this, the repressor proteins render the infected bacterial cell resistant to additional infection by other similar viruses. Next, the virus will insert its DNA directly into the DNA of the host, and it becomes a physical part of the chromosome. This is done by the fusion of two pieces of DNA. It's from the host and from the virus. From this point forward, every time the host replicates its DNA, it will also replicate the viral DNA. This creates daughter cells of the host that are also infected with the virus's DNA. It is possible that the virus will be part of the bacteria for long periods of time. This can range from generations, even extend to an eternity, forever. But eventually the phages can alter the phenotype of the bacterium. Such is the case with diphtheria and cholera, which are both caused by E. coli. And this disease it's caused by disease-evoking proteins that are found in such illnesses. It is also possible that sometime in the future, the viral DNA is excised from the host DNA, and that's done through a process called induction. After this, the virus becomes active and begins to synthesize its own DNA, and as in lytic replication, it synthesizes its own DNA and begins to assemble itself within the bacterial cell and will eventually release from the cell, and the cell will lyse. Thank you.